Hello everyone, this is Suman from DNA Bioscience. Today we are going to discuss about the type 2 hypersensitivity reaction. In the last class, we have discussed about the type 1 hypersensitivity reaction, the mechanism and all. And today we are going to discuss about the type 2 hypersensitivity reaction. So let's discuss the key points first. The antibodies which are involved in type 2 hypersensitivity reactions are particularly the IgM and or the IgG type, and it is an antibody mediated reaction. As we have discussed, the IgM antibody or the IgG antibody is involved in the type 2 hypersensitivity reaction. So let's begin the mechanism. Suppose this is the RBC cell, and we know that in the surface of RBC antigens are present. And the next step is the antigen produce the specific antibodies. Then the antigen antibody complex forms. The complements then attach with the antibody Fc region. The complement becomes activated and it results in opsonization with phagocytosis, or you can say the lysis of the red cell. So what happens? This is the surface of the RBC which contains the antigens. The antibody get attached to antigen and form the antigen antibody complex. Then the complement bind to the Fc portion of the IgG antibody and it gets activated and results in opsonization. What do you mean by opsonization? Opsonization means generally it is a process to identify or recognize the invading particle which is going to be in the phagocytosis process, or we can say it is going to be phagocytized. Then what is phagocytosis? Phagocytosis is the process of engulfment of foreign particles, or you can say the microorganisms. So the process of engulfment of the foreign particle is called the phagocytosis. In type 2 hypersensitivity reaction, the complement system is involved. So it is also called the complement system mediated hypersensitivity reaction. The next mechanism is which involve the ADCC. ADCC means the antibody dependent cell mediated cytotoxicity. ADCC mechanism, it involves the natural killer cell. The natural killer cell first recognize the antigen from the surface of the RBC. What is a natural killer cell? Natural killer cell is a type of WBC, or you can say it is a cytotoxic lymphocyte which recognize the antigen in the surface of the RBC in this case. So the natural killer cell recognize the antigen from the surface of the RBC, then it express the Fc receptor. The Fc receptor then binds to the Fc portion of the antibody and form a complex. The natural killer cell contains granules and also the opsonins which are very toxic. The opsonin is a substance which binds to the cell and they make more susceptible for the phagocytosis process. But here the phagocytosis is not going to occur. The opsonin create the pores in the surface resulting in the cell lysis. Okay, so the example of Type 2 hypersensitivity reaction is, you may have heard that disease, erythroblastosis fetalis. It is also called a deficiency hemolytic disease in case of infants. So what happens in this disease? The Rh positive RBCs of fetus. The fetus contain the Rh positive RBC, which comes in contact with the Rh negative RBC of mother. So the mother contain the Rh negative antigen and the fetus contain Rh positive antigen. So during the birth process, the Rh positive RBC and the Rh negative RBC gets combined. After that, the anti-Rh antibodies are produced, and the anti-Rh antibodies they remain in the bloodstream of the mother. So in the next subsequent pregnancy, the antibodies which are present in the bloodstream, they are released to the placenta and they result in the destruction of fetus. This fetus RBCs. So this is all about the type 2 hypersensitivity reaction. It involves the IgM and IgG type. It is antibody mediated. The macrophages and the natural killer cell, they recognize the antigen on the surface of the RBC. Then the example is erythroblastosis fetalis, and opsonization, as we have discussed, it makes the foreign substance more palatable for the phagocytosis process. And the complement system is, they are the set of proteins which work together to generally to complement the action of antibody in destroying bacteria, or you can say the foreign particle. And the phagocytosis is generally the process of engulfing of the foreign particle or a bacterial cell. And ADCC is a type of mechanism in which they contain the natural killer cell, and the natural killer cell they express a Fc receptor. The receptor then binds to the Fc portion of the IgG antibody, and they release the toxic granules which results in the cell lysis. So that's all for now. I hope it will help you.